Hello everyone, I'm Siyam Subair, a faculty at CTAS School of Engineering, Technology and Applied Sciences. This video talks about the concept of nanotechnology and the application to produce antimicrobial fabric. My research students Harnoor, Aanchal and Solomiya will take you to the laboratory demonstrations. The video is organized into three parts. The first, basic concept of nanoparticles, which is up to the standard of middle to high school and bit beyond. The next two are laboratory demonstrations. We will start with silver nanoparticles by Harnoor and then move on to nano-herbal encapsulation by Solomiya and Aanchal. Nanoparticles are tiny, but how small are they? In order to get a proper understanding, we will start with a known object. For example, full stop. The full stop is almost like 1 millimeter in diameter. If you divide that into 1000 units, that's micrometer. The respiratory droplet that we talk about during the COVID time is 5 micrometer or even smaller. It's almost the size of a red blood cell. Bacteria are even smaller, around 0.5 to 3 to 5 micrometer. Viruses are even smaller. For example, the SARS-CoV-2 or COVID-19 virus is around zero point 0.1 micrometer or 100 nanometers. The molecules are even smaller. So the nanoparticles size range from a little bit bigger than the molecule and smaller than the microbes. So what's the specialty in the size? One of the important effects of smaller particle size is the surface to volume ratio. When the particles get smaller and smaller, the surface to volume ratio increases. In this case, you can easily understand by the blue balls and the red balls. The volume would be the same, but it's divided into smaller balls. So in the blue balls, you can see the surface which is not covered, which is given in the yellow color. Those are bigger areas. When you break down into smaller balls, it covers almost bigger area. When you break down into even smaller, it would almost cover the entire surface. So this has a great application in the paint or aerosol application. Similarly, the antimicrobial particles also effectively cover the bigger surface area. Let's see the benefits of nanoparticles. As we discussed, they cover the large surface area because they can be tiny. They can also be very stable for a long time. Another benefit of nanoparticle would be the long lasting effect because we can include the active ingredient inside the shell, so that is protected from the environmental degradation and that releases slowly so that it can last for a longer period of time. Nanoparticles are used in multiple fields for multiple purposes. For example, controlled drug release in the aerosol industry, in electronics, and also the aerosols with smaller particles. We are focusing on antimicrobial particles, particularly for the PPE, personal protective equipment. At Centennial Labs, the full-length lab coat as you see here are mandatory. And in addition, we have one more PPE, which is the mask during the COVID. So these PPE are a great way of protecting us from microbes or pathogens, but they can act as carriers of those concerned particles or viruses and bacteria. So one way to get rid of those is to use antimicrobial nanoparticles. This is a prototype of antimicrobial lab coat coated by herbal particles, which are safe and effective against bacteria and viruses. This was funded by ABES Centennial College, and if you apply 1 million bacterial cells within 10 minutes, it will reduce to less than 1. Now, let's talk about how to make antimicrobial nanoparticles. There are two broad categories. One is the metallic, the other one is the herbal. In the metallic nanoparticle, silver is the most common one. There are other titanium, zinc, gold nanoparticles as well. In the silver nanoparticle formulation, We will take silver nitrate. As you can see here, this is almost like a colorless liquid. And in that, the silver would be in the oxidized state, which is given as +1. We use a reducing agent such as trisodium citrate, which reduces the oxidation state to 0. So that means the metallic silver will be formed. One of the advantage of this silver nanoparticles would be it's very cost effective. So if we apply the colloidal silver onto the fabric, that's going to be really heavy and too expensive. In the herbal nanoparticle production, we need an active ingredient. There are thousands of herbs which can provide effective protection against pathogens. We have selected oregano essential oil, which includes two active ingredients, carvacrol and thymol. Both are phenolic derivatives and effective against pathogens, particularly bacteria, fungi, and viruses. Once we extracted the active ingredients, we need a shell to protect it. So this prevents the environmental degradation and release it slowly for a long period of time. So one of the polymers that we use is chitosan. So the chitosan is a natural polymer which is biodegradable. The chitosan itself has some antimicrobial property and releases slowly. A good example is like a sponge. If you put water onto the sponge, that reduces the evaporation. But we also need the stability. To increase the stability, we use some cross-linkers which bridges the molecules so that they are stable. We have also used a synthetic polymer, which is sulfonated polyester, which work in a different way. The sulfonated polyesters, when dissolved in water, forms micelles. In the micelles, as you can see, the sulfonated parts form the head, which is hydrophilic, and hydrocarbon forms the tail, which is inside. So the micelles organized into two layers where the hydrophilic part is facing water and hydrophobic part is hidden inside. When we introduce our active ingredients, which are essential oil and mostly the hydrophobic, they go easily inside the core part and hide inside the micelle. So the micelle protects, it acts as a case and releases the active ingredient in a slow rate. Now let's move on to the laboratory demonstrations.
Hello everyone, this is Harnoorkarn, and in today's module I'm going to show how to synthesize silver nanoparticles and then apply that solution onto the mask to make an antibacterial mask. Yes, so we are going to the laboratory to basically synthesize the silver nanoparticles by boiling the AgNO3 solution and then to titrate it with trisodium citrate. So let's go to the laboratory and do the experiment. Get the materials as well as the protocols. Materials we need an earthenware flask, a small beaker, a heating plate with magnetic stirring, a thermometer to measure the temperature, some foil papers, some pipettes, a pipetter or a bubbler, a magnetic stirring bar, and for the chemicals we need AgNO3 and trisodium citrate. Step 2 is to read the protocol, do the calculations, and make the required chemicals. Hello everyone. So in this video, I'm going to show you how to make the silver nanoparticles. So first of all, as I just showed you what the materials are required, and I have made the required chemicals. So for the chemicals, we need 0.01 molar AgNO3, which is in the flask right here, you can see. And I'm heating it on the heating plate till the temperature of 80 degrees Celsius. The moment it will be 80 degrees Celsius, I will add drop-wise trisodium citrate into it to titrate it chemically. So now we can see the temperature is rising, and the moment it will be 80 degrees Celsius, I'll be titrating it with the trisodium citrate. One thing to make sure that we always do the titration inside the flame hole so that any volatile substance, the droplets and the aerosols do not come outside for the safety purposes. And we always wear a mask and the gloves for these are included in the good lab practices. So So now let's titrate with a trisodium citrate. So you can see that I do have, there are two ways actually. Either we can do it with the burette. Right now I'm doing it with the dropper. So I take some trisodium citrate into the plastic pipette with the help of the dropper, and then drop wise I will be titrating it. You can see that that's the stirring bar which is completely stirring the solution. So drop wise I will be adding trisodium citrate into the solution, and which will be mixed. You can adjust the speed of the stirrer with this one, and you can temperature can be done set with this knob, and very slowly we will be adding the trisodium citrate till all of this is being titrated. Okay, so here we can see that we are almost on to the last stages where most of the AgNO3 has been reduced with the reducing agent trisodium citrate. Now let me just give you a little bit insight of what exactly is happening inside the flask. So AgNO3 was having the valency of the Ag, that is the silver, in the metallic form of +2, and with the help of the reducing agent we are trying to reduce it to 0 so as the silver nanoparticles can be formed. And this yellow color which you can see it is developing lightly is the indication that the silver nanoparticles have been formed at the high temperature of 80 degrees Celsius and continuous stirring. I'll keep on stirring this and I will update you with the results. Thank you. So after the completion of the chemical titration, silver nanoparticle solution has been made. Now the masks can be simply put into the solution and then pressed with a hot iron to impregnate the silver nanoparticles on it. Thank you.
So first step in preparing nanoparticles is making a solution of chitosan. Chitosan is not soluble in water, that's why we are dissolving it in acetic acid, 1% acetic acid. So this is the solution that was kept overnight, and it was steering overnight, so we can see it really clear and nice. And now we will be adding twin 80 to bring all of the components together and to make it more uniform. Okay, so here we are dissolving polyester AQ48 in distilled water, and we are heating it at 60 degrees Celsius so that it can be dissolved, and hydrophobic ends can be outside and hydrophilic ends can mix with water to make micelles. So this is polyester AQ48S. We are dissolving this in distilled water and we are heating it to 60 degrees Celsius so that it can be dissolved properly. The hydrophilic ends will mix up with water and hydrophobic tail will be outside to form micelles. So now we are adding carbachol here just slowly and stirring it so that it can mix in a well way and we can get nice micelles. So to the solution of chitosan and twin 80, which we produce by heating it at 60 degrees Celsius for 1 hour, we'll be adding thymol here. And we'll be mixing it so that thymol will act as antimicrobial agent here. So we just added a stirring rod here to mix thymol properly with the chitosan and twin 80 solution, and we'll keep it for next 20 minutes to mix it up. So here we have the identical solution of polyester Q48 dissolved in distilled water, and we added thymol, which is antimicrobial, and we are waiting until it is fully dissolved, about 1 hour. So the solution is clear and slightly brownish, as you can see. So now we'll be adding 0.5% of sodium tripolyphosphate, which is a cross-linking agent, to the solution of tween 80 with hydrogen and TPP, and test well. We'll add it to this slowly to get a good matrix so that we can keep molecules in their place. The more we'll add this, the more we'll get the nice precipitate. So for preparing nanoparticles, we centrifuge the solution of TPP with thymol and titosan for 30 minutes. And now we are just separating the supernatant to get a nice precipitate, which we can wash ahead. So now when we separated the supernatant and the precipitate, we need to resuspend it with the solution of tween with distilled water in order to purify our nanoparticles even more. And afterwards we will vortex it and we will centrifuge it again. Okay, so now we want to wash our precipitate twice with the distilled water, which we have already cleaned with 1% tween 80, to get a nice and clean precipitate at the end.
So far we have demonstrated two broad ways of making the nanoparticles. Once the particles are ready, it's time to test the particles against the microbes if they are working. The way we use it, we coat the plates with the concerned microbes. In this case, you can see this the blurry area are the microbes growing, and the particles are inserted through the cylinders, as you can see, and the particles will diffuse and make a clear zones, meaning that the microbes are killed. So the bigger the diameter of the clear zone, the higher the efficacy would be. So generally, when the particles are smaller, the efficacy would be higher. So once we decide the particles are effective, then we coat onto the fabric, and that becomes the antimicrobial fabric. In this video, we didn't cover how to coat that. So there are multiple ways of coating that. And even without coating, it can be used as a sanitizer. So the particle itself is effective against bacteria and viruses. Thank you.